So it's my great pleasure to introduce Professor Tom Rando from Stanford, my mentor in the old days. So Tom, of course, I'm sure many of you actually have heard his talk here in Hong Kong. I'll just give a very brief introduction of Tom. Tom is an MD PhD, so he got his education done, both MD and PhD at Harvard, and I think you did your residency, so correct me if I'm wrong. So you did your residency at UCSF, yes, and then subsequently you moved to Stanford and start your clinician life, and also started his research lab at Stanford. Right? So over the years, of course, Tom has done incredibly in the area of muscle stem cell and stem cell aging, and so recently Tom has been elected into the National Academy of Medicine. And of course, there are many different awards that Tom has also gotten. So I will just let go the details. So I've been working with Tom for quite many years, and even as of today, he's still a great mentor. So I'm a PI, and he's still teaching me a lot of things. So I just can't thank him. So I just want to take the opportunity to thank him. Okay, so with that, so I will cut it short, and because Tom is our IAS Senior Fellow, so he'll be here from time to time. So if there's a time that you feel that you want to talk to him, let me know. I'll try to arrange, and so next time when he come visit, we can discuss with him. Okay, so with that, so please join me to welcome Tom.
My great pleasure and honor to be here and to be an IAS Fellow. And for those of you who know Tom, you'll no doubt realize that I learn much more from him than he learns from me, and that continues to be the case. I'm going to talk today about some work that goes on in our lab in this kind of intersection between stem cell biology and the biology of aging, which is where we work. We work on both, but a lot of times in between. And I'll give some background information. I'll talk a little bit about some work that was recently published first that I think is of great interest to stem cell biologists everywhere. It's very important to us, for sure. And then I'll present mostly some unpublished work in this kind of curious area of aging and rejuvenation, and some interesting data we have in this area. So let me just start by presenting an illustration of the cell cycle that you're all familiar with as cells go through G1, S, G2, and M, the traditional cell cycle. But then outside of this cell cycle, many cell populations will enter these non-cycling states, or G0 states. For example, terminal differentiation of neurons or cardiomyocytes or muscle fiber nuclei, where cells withdraw from the cell cycle permanently. There's this state related to the biology of aging called senescence, where cells that have divided many times will withdraw from the cell cycle and again be irreversibly in a G0 state. But then there's this state of quiescence, which is a very important state in the biology of stem cells, where cells are outside the cell cycle and may persist in this G0 non-cycling state for a very long time, but then be able to enter the cell cycle, divide many times, and re-enter the state of quiescence. So this is a non-cycling G0 state that's importantly reversible. And in fact, that's the whole point of it. And so what I'll talk about today is some aspects of quiescence, this interesting state, its regulation, its dynamism, and I'll come back and talk about the aging of cells in this state, which is something of great interest to us. So our interest in the lab focuses on stem cells in general, but a major focus is on stem cells from skeletal muscle, illustrated here. And even though there are many different types of skeletal muscle in the body, we really don't know what the heterogeneity of the stem cells are in all these different muscles. There are more than 500 muscles in the body. Probably the stem cells in different muscles are different, but I will refer to them generally today as muscle stem cells, even though likely they're not all one thing. This is a cross-section of skeletal muscle, in this case from a mouse. And this is a routine histologic stain. And each of these structures is a so-called muscle fiber, which is the mature form of the muscle cell that allows us to contract and move our joints. And they're one of the true few syncytia in the body, meaning they're one cytoplasm and many, many nuclei. And so here's a single, what would be otherwise a tubular structure cut in cross-section that would extend the length of an individual muscle. Now within this field, there might be one or two of these so-called muscle stem cells that I'll describe to you. And this is illustrated better in this slide, which is an immunohistochemical stain of muscle with the extracellular matrix protein laminin stained in white, shown here, the nuclei are in blue. And there's this transcription factor, PAX7, that is expressed in virtually all muscle stem cells and uniquely in muscle stem cells. So it's a very, very good marker of this population. And here's one of these such muscle stem cells shown here. And it's stained for PAX7 in the nucleus, but there's actually a small little cell sitting right here adjacent to this muscle fiber. They are also called satellite cells because they seem to be almost like orbiting this muscle fiber. Now, these cells are quiescent. They sit here in this muscle. They don't divide. We think that they can remain in the muscle of a human for decades, potentially, certainly years, without dividing. So they're really professionally quiescent cells. And this is actually an electron micrograph of one of the very first satellite cells identified by Alexander Moro in 1961. And what you see here is underneath here is this big, large muscle fiber. You just see a little bit of it here. And here's this little cell sitting underneath the basal lamina of this muscle fiber, so very, very closely opposed to this skeletal muscle fiber, the mature cell. And it's pretty much a cell that's almost entirely nucleus. There's a little bit of cytoplasm and very heterochromatic. So this is a typical appearance of a quiescent stem cell. Very tiny, mostly nucleus, and very little in the way of organelles, organellogenesis going on. Well, if these cells are sitting there in this muscle, these rare cells, they may account for maybe 1 or 2% of the total nuclear content of the muscle. What are they doing there? They're just sitting there in reserve. Well, it turns out in response to different kinds of injuries, these cells have tremendous regenerative potential, and that's illustrated in this slide. So here's uninjured muscle. Here's an area of muscle in which that structure has been totally destroyed by a toxin. And then within a few days, these cells, these very few cells, in this chaos, they begin to divide. They divide enormously. They make many daughter cells, which ultimately line up and begin to fuse with one another into these little structures called myotubes, which over the course of the next few weeks will enlarge and restore that normal structure again. So really from this chaos, restoration of this normal structure and restoration of the stem cell pool. So they can not only make differentiated tissue, but they can self-renew. So true stem cells in that sense. So this is again, back to the cell cycle. We've been very interested in this quiescent state, and these are just some publications from our lab over the years in which we really studied how this state of quiescence is regulated and how dynamic it is. So Chris Bjornson, a former postdoc in the lab, showed that this maintenance of quiescence, that the activation of the Notch signaling pathway is important to maintain quiescence. And if you block Notch signaling, these cells spontaneously exit quiescence and begin to cycle. And Tom, when he was a postdoc in the lab, published his paper looking at this microRNA 489 and its target deck, which he still works on, has some very nice work coming out on that, in which he showed that if you knock down this single microRNA, you could again break quiescence and have these cells enter the cell cycle. So it seems to be very actively regulated. Another postdoc in the lab, Ann Tang, showed that in the process of this activation, that to go from this quiescent state to this activated state, these cells had to undergo a lot of autophagic activity. So autophagy was induced and was necessary to exit the quiescent state and enter this cell cycle. And then Joe Rogers, another postdoc in the lab, showed that this quiescent state could be really quite dynamic. And he described a new quiescent state, which he termed G-alert, in which these cells can really move from a state of quiescence to a almost closer state to entering the cell cycle and more readily enter the cell cycle and more actively make muscle. So it's a very interesting state. We used to think of it as a totally dormant state, but in fact it's very actively regulated and very dynamic. And the more we learn about it, the more interesting it becomes. So it's really been interesting to figure out how to study these quiescent cells. They're tiny, they're not doing anything. And one of the ways that we've characterized it over the years is to begin to understand how they exit quiescence and begin to enter this cell cycle. And this is an illustration of how these cells go from a quiescent state into the first cell cycle by just watching them by time-lapse microscopy. So each of these horizontal lines represents a single cell that was imaged, was isolated from the animal, put in culture, and watched. And the question was, how long does it take to divide? And what you see is that there's a heterogeneity in how rapidly they divide, but these cells take a very long time to exit from this quiescent state and enter the first cell cycle. In this case, on average, almost 40 hours. Whereas if you look at the next division, the same cells, they then are now much more synchronously in terms of the amount of time, and they divide with a average time of about 12 hours and continue to do that thereafter. So there's something very interesting and very unique about this first cell division, this process from which the cells go from being a quiescent state into one in which the cells actually divide. And over the years, we've actually begun to think of this as to two stages. There are these tiny cells that the first thing that has to happen that takes many, many more hours is to go from this tiny quiescent state to this so-called activated state from which these cells can divide. And then this is just the traditional cell cycle. But this first division is very different because what has to happen is the cells have to grow, they have to undergo tremendous amount of macromolecular synthesis, tremendous amount of organellogenesis, to the point where they're actually capable of dividing. So there's a really different first division from every subsequent division after that. So how have we studied that is that what we do is we take an animal, we sacrifice the animal, and then it takes a few hours where we digest the tissue, we isolate the cells, and then we isolate and purify these cells by flow cytometry. Okay, so we've always called this the quiescent state. So we've isolated them, we have the cells, and then over the course of the next 2 to 3 days, they activate and then they divide. So what's been interesting is we, of course, study these cells so-called ex vivo. We've isolated them from the tissue, and yet we've referred to this state as the quiescent state as if it really reflects the state of these cells in vivo. Well, the question has always been, to what extent is that a good assumption? To what extent are the cells that we study ex vivo as a quiescent state compared to this activated state similar, in fact, to the cells in vivo? And so this is a question that we started to ask recently, and one of my postdocs in the lab actually took it upon herself to ask the question, can we begin to understand how similar these FACS-isolated cells, which basically stem cell biologists around the world use to study the basal state of stem cells, compared to the way these cells are actually in vivo in their native state. And she began to do this by the transcriptome, sorry. And so this is work by Cindy van Veldhoven, a postdoc in the lab, and she used this technique of TU tagging, in which basically you can express an enzyme in cells in an animal that can convert an analog of uracil into one that can be incorporated into nascent RNA. So we could express this enzyme, UPRT, specifically in muscle stem cells by breeding them with a PAX7 Cree-ER mouse. These have UPRT in the Rosa locus, so that if we give tamoxifen to these mice, they express this enzyme specifically in muscle stem cells. Now if we give this analog, TU, every RNA molecule that's synthesized in these muscle stem cells will incorporate TU into the nascent RNA. If we then purify the RNA that's TU labeled, we will have the transcriptome of the cells that were labeled in vivo. And this is really a way of asking, can we understand what the transcriptome is and what the transcriptome dynamics are of these cells in vivo? So this is just an example. So this UPRT is HA tagged, so here it's expressed in this PAX7 positive cell. And what we're seeing here is we've pulled down the TU labeled RNA either before, shown in blue, or after tamoxifen, shown in red. And you can see that without tamoxifen administration, basically none of the RNA is labeled with TU. But after tamoxifen, from these muscle stem cells, we get this large increase in TU labeled RNA. And we've looked for the specificity of this by looking at other cells in the muscle, and we look at basically the bioattenuated pulled down RNA. In the muscle stem cells, we see no RNA pulled down from the muscle fibers or any of these other mononucleated cells in the tissue. So it seems as if we're able to specifically label RNA in muscle stem cells in vivo. And we can ask, how does this compare to the RNA that we see from FACS-isolated cells that we otherwise study normally. So here's an example of an experiment that Cindy did. She just gave TU 6 hours before isolating the muscle. And then she pulled down the RNA, the TU-labeled RNA, and asked what genes are expressed in that population compared to the total RNA from freshly isolated cells. And what is shown over here, here's the enrichment of many quiescent-specific genes in these TU-labeled RNA, whereas some of the other quiescent-specific transcripts are not particularly enriched, perhaps because they're much longer lived, and they're much longer lived than the 6-hour pulse that was used to label the RNA at this point. So indeed, if we look at the RNA that's labeled in vivo, whether we pulse for 6 hours, 24 hours, or 96 hours, we see that we obtain very different transcriptome profiles. That's shown over here by this PCA analysis. So here we're seeing cells that are labeled for 1 day or 4 days are more similar to each other than the RNA from cells that are labeled for just 6 hours, suggesting that there's a lot of very active RNA that's rapidly synthesized and rapidly degraded, but there's also a lot of RNA that's stably made and stable over the course of many days. So if we looked instead at using a parallel complementary technique where we add EU, and this is now, we can do this with any mouse, we don't need this enzyme UPRT, EU is taken up into nascent RNA, but then at that point, because it's not specific to the muscle stem cells, we actually have to go through this process of isolating the cells by flow cytometry and asking, does this EU labeling correspond to what we see by the TU labeling as an index that we're actually measuring nascent RNA? And indeed, if we're looking here at this panel, we see several of these genes that are expressed in total RNA. Of the ones that are enriched by TU labeling, we've shown here, they're also enriched by EU labeling, whereas those that are not enriched by TU labeling are not enriched by EU labeling. So this just basically confirms that the TU labeling in vivo, in the presence of this enzyme UPRT, truly is labeling the transcripts that are expressed in vivo in the quiescent state. So this really began to lead us to ask the question, if the transcripts that we see from freshly isolated cells are somewhat different than the transcripts we see in vivo, can we understand what is happening during the isolation procedure? Are we actually synthesizing a lot of RNA during the isolation procedure that's giving, in a way, a bias towards what we're seeing by freshly isolated cells and looking at their transcripts? And that's what we did this experiment here. We looked again at these cells that were labeled in vivo with a pulse of TU in vivo versus where we isolate the cells and expose those cells to TU during the isolation procedure. Now, in this case, we're only labeling transcripts that are synthesized during the isolation procedure. And what we see is we look at the number of cells, number of transcripts that are differentially expressed. There are over 1,200 transcripts that are at least fourfold enriched by exposure to TU during the sort. So there's a lot of transcription going on during the sort that is not reflective of what's happening in vivo. But interestingly, over 500 genes are fourfold downregulated, suggesting that during this process that we've been using all these years to isolate stem cells, there's a tremendous amount of transcript synthesis, but also a lot of transcript degradation. Okay, so clearly our understanding of the transcriptome from freshly isolated cells has been biased by the fact that we were not taking into account all that was happening during this even few hours of isolation, transcripts being expressed and transcripts being degraded. This is just shown another way with a volcano plot. Here you're showing transcripts that are increased during the isolation procedure and transcripts that are decreased during the isolation procedure. So clearly a lot of dynamics occurring during this time that is clearly giving us a different picture of the transcriptome in vivo. If we actually look at this is the PCA analysis of transcriptional analysis of freshly isolated cells shown here versus those that are labeled in vivo and those that are labeled ex vivo, you can see that each of these actually has a strong correlation to the total transcriptome. And yet they're even more correlated with the total transcriptome than they are with each other. So there's clearly a different population of transcripts that are being synthesized and a different population that are being degraded to give us this freshly isolated population. Another way to ask this question is we use a transcription inhibitor, alpha-mannitine, during the sort procedure, and if we do that, then we're blocking this upregulation of transcription that's happening during the sort. And indeed, several thousand genes now show a downregulation because instead of being synthesized, they're not being synthesized. But surprisingly, there are also a population of transcripts whose levels are increased in the presence of a transcription inhibitor. So again, this suggests there's a lot of dynamic interaction between what's being synthesized and whether there's a net increase or a net decrease in the population of specific transcripts. So finally, as a way of asking this question using a completely different approach, and Tom Chung has also pioneered this in his own lab, is we took an approach that actually will be very useful for stem cell analysis among different tissues, and that is to ask the question of what is the transcriptome in vivo of a population of quiescent cells. We in fact fixed the tissue in vivo and then did the sort and asked what the transcriptome is. So we didn't know if this would work, and we've been trying this both for transcripts and for protein, but if it's possible to do some kind of light fixation and then purify cells such that they're not changing during the isolation procedure, this would also give us a much more accurate picture of the state of the transcriptome or the proteome in vivo. So we fix cells, and then we ask, how does this transcriptome of the fixed cells compare with the transcriptome of unfixed cells the way we've been doing traditionally? And there are two things to look at here. First of all, almost 10,000 transcripts are unchanged whether we fix the cells or don't. But there are some over 4,000 genes whose levels are increased if we look at the fixed cells compared to the unfixed cells, and some 3,000 plus genes in the other direction. So these are genes that are induced during the isolation procedure, and these are genes that whose transcripts are reduced during the isolation procedure. So it really, again, shows to the importance of understanding what's happening during these very simple enzymatic and mechanical dissociations where you're actually isolating cells from in vivo to the point where you're going to do your omics analysis, and in fact they're quite different from one another. So getting back to the fundamental question we were asking is, how similar are these cells that we've been purifying by FACS compared to in vivo? It's sort of a question of, is the glass half full or glass half empty? On the one hand, most of the transcripts in this population actually do reflect the in vivo situation. So to the extent that one can look at this FACS-isolated cells and say, how does this transcript reflect the in vivo state? For the most part, that's true. And yet, it's important to also understand which transcripts are induced during this process and which transcripts are reduced during this process that are unique to that population and that purification procedure that would give a somewhat biased view of the transcriptome or the proteome from freshly isolated cells. So this has been important for us, and I think it's important across the field to understand that the process of isolating stem cells and analyzing them is actually biasing our results compared to the way we can study this in the in vivo niche. So we're trying to understand more and more the in vivo transcriptome and the in vivo proteome better and better than we've done using this FACS isolating technique over these years. Okay, so I wanted to present that. That was recently published in Cell Reports, and we've been pursuing this in more detail to try and expand our analysis of the in vivo state. But I'd like to now turn to the question of the aging of stem cells and this issue of how stem cells age and can they be rejuvenated. So we've been very interested for a long time in how stem cells in a young individual versus an old individual are different. Clearly, the older we get, the less we're able to repair our tissues from our stem cells, whether it's bone or skin or muscle. And our interest has been, can we understand this loss of regenerative capacity in terms of changes in stem cell function? So we've thought of this in several ways, and here I've illustrated what I consider two different ways in which stem cells age. So there's a conventional way called replicative aging, in which you put cells in culture and you let them divide, and eventually they reach the senescent stage. But during this process, they undergo what is considered replicative aging. So the more they divide, the kind of older they get in a replicative way. But at the same time, for these populations of cells like these muscle stem cells that exist in a quiescent state in vivo, over time, they will just age as any non-dividing cell in the body ages. So like a neuron or a cardiomyocyte. Over time, those cells change, as will a quiescent stem cell that is still capable of dividing, but is existing and persisting in a quiescent state. So we've been very interested in understanding this kind of chronological age of quiescent stem cells. And the reason is because of the difference in the ability of these cells to repair tissue in young versus old, in this case, mice. So here we're looking at the cross-section of muscle from a young animal, and then regenerated after a first injury and regenerated after a second injury. And these muscles can be injured over and over, and you'll get very, very good regeneration over and over. With age, that situation changes. So here's an old muscle. After a single injury, you already see a lot of this area of incomplete regeneration, essentially fibrosis. After a second injury, you see much more impaired regeneration. So clearly, the functionality of these cells in old versus young is very different. We know that this is not due to a loss of stem cells. There's a perhaps 20 or 30% decline in number, but not nearly enough to account for this marked impairment of the regenerative response. So a question we've had over the years is this age-related impairment of regeneration due to the aging of muscle stem cells? And we've done this by looking at both molecular and biochemical readouts as well as functional readouts. This is an old, actually microarray analysis of the total transcriptome of old cells versus young cells, and you can see that actually they really are quite similar. There's not a tremendous change in the transcriptome in going from young to old, but they're sufficiently different that by PCA analysis you can clearly distinguish young cells from old cells. So there are transcriptional changes, even though they're subtle, but we've looked at a lot of other basically signatures of stem cell aging, and one of them that we found, in particular this work was done by Ling Lu, a senior scientist in the lab, looking at the epigenetic profiles of young versus old cells, and what she found for these muscle stem cells in particular is this mark that we've been very interested in, which is this repressive histone mark, H3K27 trimethylation. You see now across transcription start sites is very little enriched in the young, but with age you see this huge increase in this H3K27 trimethylation mark. So this is a sign of gene repression, and it increases dramatically across the genome in these old muscle stem cells. And we've been interested in what the acquisition of this repressive mark means for stem cell function, and we've been focusing on several gene or gene families in which you see an increase in this repressive mark as well as a decrease in the transcriptome of those genes. One in particular that we've been interested in is these histone genes themselves. So this is a look at histone gene cluster here, and you see we're looking at this activating mark, which is H3K4 trimethylation, and this repressive mark, H3K27 trimethylation. You see very little of this repressive mark in the young, but in the age you see a large increase in this repressive mark across this gene cluster. So again, an increase in this repressive histone mark, and it's associated with, as we look at these histone genes, a decrease in transcription of each of these loci. So this is an interesting profile, and we're trying to understand what this means in terms of the function of these cells. One interesting thing is if we look at the protein level, there's also a decrease in these histone proteins in old cells compared to young cells. So it's turned out to be, I think, very interesting as we look at the distribution of histones across the genome. We see large areas of histone-free regions in old cells versus young cells. And so we're pursuing that now in terms of understanding what that means in terms of the function of the cells. Is it related to DNA damage susceptibility? Is it related to changes in gene expression? But we think this is going to be an important signature of aged stem cells in muscle, and we think in other stem cells as well. But what about functionality? So those are some molecular readouts. Are there functional changes that we measure in old cells compared to young cells? And that's what I want to spend the rest of the time talking about, is some interesting data we have on how old cells behave differently from young cells in this regenerative process. Now, I mentioned that there's a large growth phase during the activation of quiescent stem cells going from quiescence to activated, and I showed this slide already of the long time it takes for the cells to exit quiescence and enter the cell cycle. Well, if you look at this in old cells, you see an even more extreme case of a prolonged time to first division. So this is the young and this is the old, and you can see in this case it's up over 50 hours for the cells to enter S phase from the quiescent state. So really the question is, are these cells now, in the old cells, in a deeper state of quiescence? I've just illustrated this here. If the young quiescent cells have to go from a very small state to an activated state over this prolonged period of time, are the old cells even more deeply quiescent, requiring even more time to exit quiescence and enter the cell cycle? So we've been studying this, and it turns out this seems to be the case by all the criteria we use to measure the functionality of these stem cells in going from quiescence to activation. Here we're looking at cell size. I mentioned they have to grow enormously. The young cells grow much more quickly than the old cells, even though they start off at basically the same size. If we look at entry into S phase by EDU incorporation, the young cells do so much more quickly than the old cells. The means are calculated here. Again, looking at this time to first division, the young cells undergo cell division much more quickly than the old cells, with again the means plotted here. And if we look at actually the aspects of this incorporation in vivo, so we do an injury to muscle, we give the mice either EDU or BRDU, we see again a much higher fraction of cells have entered the cell cycle at this point at 48 hours in young versus old. So there seems to be this marked delay in the cell's ability to exit quiescence, enter the cell cycle, and engage in tissue repair. So we think that a lot of the defects that we see in the regenerative response is because of this deeper state of quiescence of the aged muscle stem cells. And the question is there anything we can do to improve aged muscle repair to make it repair muscle much more like the young stem cells can? Well, over the years, we've looked at a couple of ways to do this. So this is work from Irina Conboy many years ago when she was a postdoc in the lab, who found that the Notch signaling pathway declined with age in these cells and was important for this process of going from quiescence to activation. And what she found is that if she could actually restore Notch signaling in old muscle by using a pseudo-ligand of the Notch pathway, the regeneration of the old tissue would go from a very impaired regeneration to very normal, young-like regeneration by just adding this one ligand for this one pathway that we knew declined with age. So just restoring Notch signaling was enough to restore the youthful functionality of old muscle stem cells. Well, through a series of other experiments, we actually looked more generally at the environment in which these old muscle stem cells engage in activation and engage in repair. And we hypothesized from that information that really these old cells are just exposed to an environment, a circulating environment that's much more suppressive than you see in young animals. And to test this, we use this a technique called heterochronic parabiosis, where we connected two animals together, either two young animals together or two old animals together as controls, or these experimental animals where we connect a young animal to an old animal. Now, these parabiotic pairs result in a single shared circulatory system. So these animals, over the course of several weeks, now you have a young tissue that's exposed to the circulating environment of an old animal, and an old tissue that's exposed to the circulating environment of the young animal. So the question is, if old tissue is exposed to the young circulating environment, what effect does that have on the old tissue? Can you modify the environment in which stem cells reside and activate to induce these aged cells to behave more like young cells? In fact, that's exactly what we saw. So in these so-called isochronic pairs, so young to young and old to old, you see either good regeneration or poor impaired regeneration, just like in the unpaired animals. But in these heterochronic pairs, the old partner, now exposed to this circulating environment of the young animal, showed basically youthful regeneration. So it suggests that these old cells, these old stem cells, can essentially be reprogrammed by the exposure to this young environment to become much more youthful. And we could see that here we're looking at, again, Notch signaling and Notch gene expression. Here's the normal young situation. Here's the suppressed old situation. And in these heterochronic situations, we see the old animal now has much higher level of this Notch ligand signaling to much higher levels of Notch signaling. So the heterochronic parabiosis allows this Notch signaling to actually be restored to a more youthful level, perhaps accounting for this improved regeneration. And over the years, several other groups have looked at other tissues and shown that in this heterochronic parabiotic paradigm, that old cells and old tissues respond to this circulating youthful environment to improve repair of a lot of different tissues, whether it's neurogenesis in the brain, spinal cord injury, pancreatic beta cell activity. So all of these functions that decline with age begin to improve again toward a more youthful level when the old tissue is exposed to this young circulating environment. So the question is this really rejuvenation? That's the working hypothesis is actually these are cells that have gone from young to old, and now through this exposure to a young environment, we're actually restoring them to a more youthful state. And it's kind of a major focus of the lab is can we identify the molecular signatures to say that we actually have young cells, old cells, and rejuvenated cells, with the rejuvenated cells being very much like the young. And we have data both from a transcriptional level and from an epigenetic level that this exposure to the young environment truly is restoring youthful phenotypes, youthful molecular phenotypes to the aged cells, suggesting that this process that really may be possible to revert or reprogram some of these aging phenotypes to a more youthful phenotype. So we're very interested in that. We're interested in what are the factors in the circulation that might be mediating that. But another question we've had over the years is, other than doing parabiosis, which you can't do very well with humans, are there more physiologic ways to rejuvenate aged muscle stem cells? Are there ways we can intervene in an aged animal to restore youthful functions to these old cells? So we've been very interested in the biology of exercise and become more interested recently. I love this article from the New York Times this year because it suggests that if you run for an hour, you add 7 hours to your life. So that just means you should just keep running, I think, because if that's true, you can just extend your life forever. So I'm not sure it's true. But the question really is we know that there are these tremendous health benefits of exercise. And they clearly have these benefits that are important in terms of health span and lifespan. And our question was, does exercise rejuvenate old muscle stem cells? So we have this idea that if we put old mice on these running wheels, we can measure how far they run and ask to what extent, if they run, do we see improvements of function of different tissues, but in this case of muscle and muscle stem cells. And this is work primarily by an MD PhD student in the lab, Jamie Brett, who found this interesting observation that led us down this path. And this is the observation that Jamie found, is that if we look at, so here's young without exercise, young with, old without, old with, and we're measuring again what I mentioned before is if you look at the regeneration of an old muscle, you see these areas that are not regenerated, so there's increased area fibrosis. So if we measure the amount of regenerated area, here's young and here's old, so it goes down with age. If we exercise the young, we see no improvement, but if we exercise the old, we see a marked improvement in regenerative capacity of an old tissue. So we've been working on muscle regeneration for a long time and exercise for a long time, but we never. So this is running for 3 weeks, and so it's not one day, unfortunately. And they run about, these mice run a lot. They actually run like 5 kilometers a night, so which is a lot for a mouse. So they love to run. They love to run. So this is at the end of the running time. We injure the muscle, we look at the regeneration. It was really surprising finding of how much better the muscle from an exercised animal has regenerated. So this really started us down the path of asking, what is the benefit of exercise, and is this being mediated through effects on the muscle stem cell? Indeed, if we isolate the stem cells and look at their functions, we see more youthful phenotypes of these aged cells now. So here we're looking at entry into the cell cycle, EDU incorporation. Here's the young level, here's the old level, and here's the old level with exercise. So improvement in terms of cell cycle entry. If we look at all of these different parameters of cell behavior, motility, DNA replication, time to cytokinesis, clone formation, they're basically all restored to some extent by this bout of exercise. So Jamie asked, well, can we begin to understand what's different about these cells now that have been exercised? And so she did just an RNA-seq analysis. And what was surprising was, well, many genes go up, many genes go down, but one gene was dramatically changed in response to exercise in these muscle stem cells, and that's Cyclin D1. So Cyclin D1 obviously has a historic role in the cell cycle. I've illustrated this here. So typically, transition from G1 to S is inhibited by RB, but in the presence of mitogens, Cyclin D1 in binding to CDK4/6 inhibits RB, which then releases the inhibition of RB and allows cell cycle progression from G1 to S. So clearly Cyclin D1 is important in terms of the kinds of cell cycle analyses that we've been studying So we were interested in really whether the Cyclin D1 can account for some of these kind of changes we're seeing with age and then in response to exercise. So first, to be sure that these changes in Cyclin D1 are not due to any a few cells that change, we looked both at the single cell level. Here looking at Cyclin D1 in terms of transcript level. So this is CT value. So this is actually higher levels in the old with exercise compared to the old without exercise, no change in GAP-DH. So an induction of this transcript, not just in a few cells, but actually in the whole population. And if we look at single molecule RNA fish, so the actual number of molecules of Cyclin D1 in these different populations, so here's young without and with exercise, Cyclin D1 levels decrease in old, but then are increased again with exercise. So this is, again, not just a few cells that have a very high level of Cyclin D1, but seems to be a change in Cyclin D1 levels across the whole population at the transcript level. Well, if we look at the protein level, we also see that here's young levels of Cyclin D1 very high, low in the old, and then partially restored again with exercise. So not only at the transcript level, but at the protein level, we see a normalization, or I guess a more youthful levels in response to exercise. Looking at Cyclin D1 by immunostaining, you see the same thing. Can't see it too well here, but expressed in the population of young, much lower in the whole population of old, but then restored toward a more youthful level, and this is quantified here in response to exercise. So it seems as if this protein decreases with age and is responding quite dramatically at the transcript and protein level in response to exercise. But is it really important in terms of mediating this transition from quiescence to activation? So we asked that first by just using an siRNA approach. If we knock down Cyclin D1 compared to the control, and we look at, again, progression into the cell cycle by EDU, if we knock down Cyclin D1, we see a decreased percentage of cells that enter the cell cycle at any one time. So it seems as if it's important in terms of mediating this transition from quiescence to activation. But this is a challenge to do because this is all done in culture. So we generated mice in which we could actually modulate or have different levels of Cyclin D1 be expressed in the muscle stem cells. And we did this by crossing a mouse that has the D1 allele floxed, again with this PAX7 Cree-ER mouse, to create mice that when we give tamoxifen, we either have wild-type levels of Cyclin D1, they're heterozygous for Cyclin D1, or they're full knockouts for Cyclin D1 in the muscle stem cell compartment only. So if we look at these populations at the transcript level by PCA analysis, here are young cells, here are old cells, here are the Cyclin D1 knockouts, and here are the Cyclin D1 hets, sort of intermediate in between. So at least at that level, they sort of phenocopy this change with age of going from wild type to less Cyclin D1 to no Cyclin D1 in the knockouts. If we look at the protein level, basically we see the same thing. So here's the wild type, here's the heterozygote, and here's the knockout. These are quantified here. So we are indeed using this genetic approach able to reduce the level of Cyclin D1 in this population by about half and then completely in the knockout. And indeed, if we look at the functionality of these different populations, we see that the reduction in the ability of cells to enter the cell cycle correlates with this level of Cyclin D1. So here are the wild type. The hets have a reduced entry in the cell cycle by EDU incorporation, and the knockouts have an even slower entry in the cell cycle by EDU incorporation. If we look at the effect of exercise, we see that just like in the old animals, the effect of exercise could increase the Cyclin D1 levels from about half in the hets to back wild type, and of course no effect in the knockout because it's not going to induce Cyclin D1 in the knockout. So looking at the hets, it looks as if the ability of exercise to induce Cyclin D1 in the old cells is mirrored by its ability to increase Cyclin D1 in the heterozygous mice. And if we look at the again the functionality in terms of EDU incorporation or cell cycle entry, again we see the wild type with and without exercise, the heterozygote with and without exercise, and the knockout with and without exercise. No effect in the knockout, but again, the heterozygote, which is markedly reduced in terms of cell cycle entry, its ability to, or the ability of exercise to induce cell cycle entry is like what we saw in the old cells. So it's as if the heterozygous is really phenocopying the old, that exercise can induce both the level of the protein as well as the functionality of the cell to enter the cell cycle. Okay, so I've been talking about this, but keeping, there's one point that I haven't made, and that is I initially talked about Cyclin D1 as mediating this G1 to S transition. But as I said from the beginning, really what we're asking is a transition from G0 to G1. And so the question is Cyclin D1 having kind of a similar role in mediating this transition as it is in mediating this transition? Well, it turns out it seems as if it's probably not. And the reason why we think that is because if we look at this canonical Cyclin D1 binding to CDK4, it's clearly binding to CDK4 in these proliferating cells. If we pull down Cyclin D1, we pull down most of the CDK4. But in the quiescent cells, if we pull down Cyclin D1, almost all of the CDK4 remains unbound. So it seems as if Cyclin D1 is playing a role in this quiescent population that's independent of the conventional CDK4 binding, conventional inhibition of RB. So we've now started looking, what I'll spend the rest of the time talking about is, what is Cyclin D1 doing in a quiescent cell? How is this acting if it is not acting by binding to CDK4? Well, if you look in the literature and you ask what are the proteins to which Cyclin D1 has been reported to bind, you find enormous numbers of proteins. So there's really a tremendous number of pathways and proteins that Cyclin D1 has been proposed to act in a non-conventional CDK4/6 independent way. So we took a bioinformatic approach, and looking at this paper from 2010, which showed Cyclin D1 as a regulator of transcription and bound to promoters across the genome, we looked at the population of genes that are, quote, Cyclin D1 targets, and we asked, how many of those genes are enriched among genes whose expression decreases with aging, and how many of those are enriched among genes whose levels are increased with exercise? So these are the two parameters we're looking at, which is decrease with aging and increase with exercise, and are also Cyclin D1 targets. And when we looked at those and we asked what transcription factor motifs are enriched, it turned out there are very few. So actually we pulled out 5 motifs, 2 of which are TGF-beta transcriptional regulators, and 2 of which are NF-kappa B transcriptional regulators, and then there's EBF1. So we've been focusing on the TGF-beta pathway as well as the NF-kappa B pathway as potentially important in terms of mediating the Cyclin D1 effect. And in fact, if you look through the literature, Cyclin D1 has been reported to be a transcriptional co-repressor of both of these pathways, the TGF-beta pathway and the NF-kappa B pathway. So we've been using inhibitors of these pathways to ask, can we overcome the effects of aging and the effects of Cyclin D1 decrease in the genetic models? So the way we do these experiments is we give the inhibitors, in this case I showed TGF-beta1 inhibitor, several days to these mice. We isolate the cells and then we ask about this ability to transition from quiescence to activation. What we see is, if here's the vehicle treated, both NF-kappa B inhibitors shown here, even more so the TGF-beta inhibitor, can really restore these old cells back to a more youthful state as if they're basically phenocopying the effect of exercise. And we're seeing the reduction of TGF-beta really restoring youthful activation kinetics. But when we look at young cells, we see the effects of these inhibitors are basically non-existent. So it seems to be truly an effect on the old cells and not just an effect in general on inducing cells to increase or to engage in the cell cycle much more rapidly. If we look at the knockout, we see similar results. So here we have the knockout cells. If we inhibit NF-kappa B, shown here, we see a little bit of increase. But if we inhibit TGF-beta, we again see a more wild-type level of activation compared to the vehicle treated. So these, again, Cyclin D1 complete knockouts, but still these inhibitors are able to improve the cell cycle entry kinetics compared to the wild-type cells. Okay, so this is our current model, and we're sort of working through this. But the idea is that in young cells, you have these cells in G0, high levels of Cyclin D1, they're inhibiting any of this NF-kappa B or TGF-beta activity, and so these cells are much more rapidly able to exit quiescence and enter the cell cycle in response to stimuli. With age, but also with the Cyclin D1 heterozygotes, we see reduced levels of Cyclin D1. So the idea is that the data suggests there's an increase of NF-kappa B signaling, TGF-beta signaling, which are actually suppressing the quiescent state and requiring longer for the cells to exit quiescence and enter the cell cycle and activate. And then with exercise, we see that that's able to restore Cyclin D1, thereby inhibiting these suppressors of activation and allowing cells to activate better. So the idea is that exercise is actually intervening in this Cyclin D1 and these inhibitory pathways to restore cell cycle activation. And we see when we exercise, again, this much improved regeneration as expected by this improved activation kinetics of these cells out of G0. So again, we think that in the same way we were seeing with the parabiosis, exercise truly is restoring youthful properties to the AH stem cells. So they go from a high Cyclin D1 to low Cyclin D1, and exercise restores the Cyclin D1 levels to these old stem cells, allowing them to have more active kinetics and more effective regenerative capacity. So we're digging down, trying to understand what the downstream targets are of both the TGF-beta and the NF-kappa B pathways that are important really for these changes in cell cycle activation and this change in the dynamics of the G0 quiescent state. Okay, so let me just end there by again acknowledging the people who did the work. So most of the work on the TU that I mentioned, TU labeling, I mentioned early on, Cindy van Veldhoven, and then all this work on exercise and Cyclin D1 is done by Jamie Brett. Antoine Demore did a lot of the single cell and single molecule analysis, looking at Cyclin D1 as well as some of the other genes in these cells. Mention a couple other people. So we had 3 visiting scholars, Mika Ikeda, Ingrid Egner, and Louis Parandini, who also worked on this exercise and Cyclin D1 project while they're in the lab. And a lot of this work is done in collaboration with our colleague at Stanford, Tony Weiss-Corey, with whom we're also working on the role of exercise in rejuvenating aged neural stem cells. Also want to acknowledge our funding support. A lot of this comes from the Glenn Foundation for Medical Research, has funded a lot of our aging work, as well as the National Institute on Aging. So with that, I'll stop, and I'll be happy to take any questions. Thank you.
Rando, thanks for the nice talk. So the question, actually I have 2 questions about the Cyclin D1 part. The first question is that when you measure the Cyclin D1 level, either mRNA or protein, are you measuring the cells that are isolated, freshly isolated, or the cells that are, well, the true quiescent cell, that whether it's a fixed cell?
So it's a good question. So all of this is done with freshly isolated cells, but we've gone back using our TU data to confirm that the level we see in the freshly isolated cells is the same, basically the same as we see in the in vivo cells, like I mentioned at the beginning. It's a good question. Yeah.
Okay. And the second question is, what do you think? It's like a general question. So what do you think about the link between the exercise and the Cyclin D1? So what does the exercise cause?
So we don't know. I mean, so we're beginning to. I can tell you one thing we know, and that is, if we take plasma from exercised animals and give it to exercised old animals, and we give it to non-exercised old animals, we can recapitulate these effects of exercise. So we're looking now at what the proteins in the blood are that change with exercise that might be mediating these effects. But we don't have any data yet to report.
So that means the exercise somehow changes maybe the whole body metabolic status?
I mean, we know certainly exercise changes the whole body metabolic status. I mean, what we don't know is what are the mediators of this particular effect. And I think it's going to be complicated for sure, but at least we know that it's activity that's contained in plasma, so we can begin to analyze the plasma and ask, can we reduce that to something that could be, potentially targetable.
Okay, Thank you.
Yeah.
Thank you for your nice talk. So I want to ask about the fixed isolated cells. So when you compare the cells that are from fixed soil and the fresh isolated cells, and we observed that around 1,000 of genes actually changed during the isolation. So I want to know your comment on that. So is it actually coming from a global effect of the cell transcriptome change, or actually just a small population of the cell-like cells that have dramatically changed during the isolation?
So I see. So looking at the genes that go up or down, is that yeah? So we've done some single cell RNA-seq, and impression we have is this is global. So we don't see any small population that showed dramatic changes. We see in the single-cell RNA-seq, we see comparable changes in the whole population. I mean, there's always a big distribution, of course, but we don't think this is related to the question you asked, is this a small population that shows dramatic changes? Rather, it seems to be a global change across the whole population. Yeah.
Okay, thank you.
Thank you for a wonderful talk, and I'm really interested in the part of the fixed facts. And your group used method to fix the cell, so I'm interested in which kind of chemical or physical method you were using.
So we use paraformaldehyde perfusion fixation.
So after that, you do the RNA-seq.
Yep.
I also see there are some genes upregulated or downregulated. So did you check the geoterm to see if they are functional related or they're just random regulated?
So we've certainly done the Go analysis, and there are terms that come up in both cases. But as you dig down into the genes, there are only a few Go terms that make sense to me. So a lot of them are just see things like certainly we see increase in heat shock genes that go up with isolation that don't if we fix, right, or immediate early genes. But a lot of the terms I think are conglomerate terms that don't really tell us too much about what the key genes are that are changing. So I think we've dug down into this a lot to try and figure out what these key genes are. I would say the Go analysis has not been very helpful. Yeah.
And is it possible that after the cell is fixed and they die and the mechanisms for protecting the transcriptome just broke and the degradation just happened randomly, and so that's why the geoterm doesn't tell much information?
I guess what I would say to that, so I'm not sure I understand what you mean, but if we look at the fixed facts data, right, so these are the transcriptome from cells that have been fixed in vivo, and we compare that to the transcriptome that we obtained from the first method we use, the TU method, in which there's no cell death, there's no fixation, we see comparable genes that are changing. So I think you're suggesting an artifact of the fixation. I'm not sure what the artifact you're suggesting is, but because those two data sets overlap, we think that the fixed facts is giving us an accurate representation of the in vivo transcriptome.
Thank you for a wonderful talk. I have a question about after 3 weeks exercise of the mice, are there any improvement in their cardiopulmonary functions? I wonder after the exercise, because for human beings, our heart function will be much stronger. So is it possible that our blood oxygen level will also increase, which will change the microenvironment of the stem cell?
I would say there are probably many, many changes that are occurring physiologically in response to this exercise. And so I wouldn't exclude any of those as being important in terms of the phenotypes that we're seeing. But I would also, again, say that the fact that we can transfer the effect by plasma transfer suggests that there's something relatively stable in the plasma that is mediating the effect. That doesn't mean that there aren't important effects of changes in hemodynamics and other metabolic changes that are associated with exercise that might also be important.
Thank you very much.
Thanks, Professor Rando. Very interesting work about exercise and muscle. I think I probably need to go to the gym after the seminar. So my question is, how long-lasting is the effect of the exercise? Like you mentioned that your mice exercise for 2 to 3 weeks, then you do the muscle injury. I'm wondering if you wait for a while or let the mice run when they were young and do the experiments when they are old, or do you think it's a transient effect or it's long-lasting? And what about the intensity of exercise? If you get your mice run for 5 kilos every day, if they run less, or what are these effects?
So again, good question. So these are all doable experiments. They take a long time. So we've done very little in terms of varying the intensity of exercise. So first of all, these are voluntary wheel running, so we're not doing treadmill, forcing them to run. So that's one caveat of this, is that the amount of exercise among the mice varies. Some of them run more than others. But I don't have any data on intensity of exercise. We have done studies, though, looking at how long the effect persists after the exercise stops. So these are 3 weeks of running. We do the analysis usually right after that. But if we wait over weeks after the exercise stops, we see a reduction each week over about 3 weeks until it's back to baseline. So the effects are not rapidly degraded after the exercise, but they're definitely transient.
And are the changes in the Cyclin D1 expressions and the cell cycle only observed in the trained muscles?
Only in the what?
On the trained muscles, because for example, you are in the running wheels only for the
Okay, so
muscles.
we've not looked at this. The problem is it's hard to figure out what muscles to use. The only muscles I can think of that we could truly consider not trained would be like jaw muscles, right? Because pretty much all the limb and the diaphragm will be used in the. So we've not looked, but it's a good question. I mean, we haven't looked at that. Yeah.
Maybe they eat more.
Maybe they eat more. Maybe no muscle is spared. It's true.
So Tom, I also have a question about the Cyclin D1 levels in the young. I mean, if you run the young mice, I mean, do they even go up?
They
higher?
do not. So you know, the way we think of this is almost as if the young cells are already kind of maxed out in terms of their functionality and Cyclin D1 levels. So no, there's not a further increase in Cyclin D1. So again, we can't quite explain this, but we see, interestingly, very little effect of exercise in any way on the phenotypes that we measure molecularly or functionally in the young. We only see it in the old.
So another question is, if you immobilize the mouse, what will happen to the Cyclin D1 level?
It's a good question. We've not done it. It's a good question. Yeah.
Okay, so if there's no further questions, so please join me to thank Tom for a wonderful lecture.